Welcome to 8.6, gene expression and regulation. Now we've talked about the translation process, the process by which the amino acid is being formed, which leads to the formation of protein. But I want you to know that sometimes the process of transcription and translation is a process which continues every time, it doesn't stop. So what would happen is if you continue to make protein and make the ones you don't need, you're going to suffer two things. One of it is you're going to run out of the raw materials which is needed for the production of protein. And second thing is you're going to be using too much energy which you don't need to use for the production of the protein, which you don't actually need. So this is the reason why gene expression or production of protein has to be regulated. Gene expression is carefully regulated in both the prokaryotes and the eukaryotic organisms. Remember the difference between the prokaryotic organism cells and the eukaryotic cells. The prokaryotic cells are the cells with no nucleus and that does not have membrane-bound organelles. The organelles have no membrane, which is covering them. While the eukaryotic cells, they are the cells which have nucleus and also they have a membrane-bound organelle in them. Now, the prokaryotic cells turn gene on and off by controlling transcription. The prokaryotic cells, they know that the process of the DNA replication, transcription, and translation is going to continue without stopping. Let me give you a good example. Let's say you have your mom bought food at home and you're on vacation for COVID-19. You started eating all this stuff. You ate all the stuff, the ones you don't need, you just eat all of it. What would happen is, one, you're going to have either constipation or you're going to have diarrhea. You're going to have to be going to the washroom, going to stool all the time, or you're going to have to run your mom into debt. Your mom is going to run out of cash because she doesn't have enough money. You're eating all the food in the house. So this is exactly what happens. So the gene knows that if it continues to let this process of translation and transcription to happen without being controlled, they would run out of resources and they're going to exhaust too much of energy. So, during the process. So we got something which we've got some materials which are called regulatory materials. One of them is a promoter. The promoter is a DNA segment that allows the gene to be transcribed. So what do I mean by this? Is a DNA segment. It's a long DNA segment which allows the process of the transcription to happen. So we can describe it like a long DNA sequence like this, whereas it's actually been segmented. So that is a promoter. And remember, the promoter is the one that actually shows the position where the transcription process is going to start. Now the operator is the one that works as a switch that turns on and off the process of the transcription. While the operon, it includes a promoter, it includes the operator, and also includes one or more structural gene that code for all of the protein which is actually needed for the job to be done. A very good operon which is known among the prokaryotes is called the lac operon. The lac operon is got from lac means lactose. The lac operon is one of the first example of gene regulation which was actually been discovered by the scientists. The lac operon is known to have three genes, like I mentioned to you before. It has three genes that code for an enzyme which is going to help to break down lactose. We know lactose is the kind of sugar which is got from milk, and we know a very popular enzyme that breaks down the lactose is lactase. When you have ASC behind it, it's going to be an enzyme. But when you have we have OSC behind it, it's going to be a sugar. So lactose is a sugar which is actually found in milk, and it's being broken down by lactase. So when this organism, when it has lactose in its body, it's going to it's going to allow the production of lactase or some other enzymes that break it down. But when there is no lactose which is present in it, so the lac operon will work as a switch which helps to turn on and off of the process of transcription. So the lac operon is off when lactose is not present. So when lactose is absent, what would happen is the lac operon is going to be turned off. But when lactose is present, is going to be turned on. The meaning is when it turns on, it wants the production, it wants the transcription process to continue to help to produce an enzyme, which is a protein, which would help to break down the lactose in the cell. So this is an example of the way it works. I'm going to explain this to you. This is a DNA, and this is a gene for the enzyme that is going to digest or going to break down lactose. This is called the operator, which is a switch that actually works here, and this is called the promoter. So what does this do? The promoter would show the RNA polymerase. The RNA polymerase is the enzyme which actually help to begin the process of the unwinding of the RNA for the transcription process to start. So the DNA wants to unwind. So the unwinding is going to start by our RNA polymerase, which is going to locate the origin of the DNA where the unwinding process is supposed to start. So when the RNA polymerase when it forms a complex, this is a complex for RNA polymerase. This is a promoter which tells you the origin or the beginning where of the transcription process. It's the one that actually shows the position where the DNA can be unwinded, and that's where the RNA polymerase forms a complex in. So to start the process of unwinding the DNA, so that RNA can be formed from it. But this guy, which is called the operator, which is a switch, is going to stay behind the RNA polymerase to block the process. So it blocks the process because there is no need to make an enzyme which is going to digest lactose, because lactose is now present in the cell. So why would you need to waste resources to make it when it's not there? So this is the way it happens. So this guy is a blockage, is a repressor, which is not going to allow the process to proceed further. But when there is lactose in the cell, or in the gene, what would happen is the lactose would go and bind with this. It binds with it like this. So when it binds with it, it is going to make it fall off from the position it is here. So when that happens, then you can see the RNA polymerase is going to proceed further. So by unwinding the DNA. So when the unwinding process continues, then a new mRNA is going to be made from the DNA. So the mRNA is having molecules which is going to be the one to be translated to help to digest the lactose, which is in the DNA. Now let's talk about eukaryotic cells. All of the ones we mentioned before were for the prokaryotes. Now, but in the eukaryotic cells, they regulate at many points, because the difference between the prokaryotes and the eukaryotes is the prokaryotes are most single cells or single cell organisms, but the eukaryotes have several cells in them. So the meaning is there might be a transcription process happening in different positions in the cell. So the materials which are needed to allow the transcription process to happen in the eukaryotic cells, they are enhancer. Another one is called a Tata box. So these are called transcription factors, and this is the RNA polymerase. So the transcription process is actually being controlled by the regulatory DNA sequence and the protein transcription factors. What the enhancer does is it's the one that actually controls the speed of the transcription. It slows down the other way, and also helps to fasten it. It either slows it down or makes it happen very fast. Why the Tata box is called Tata box? Because it contains 7 nucleotides which are responsible for the production for the transcription process in eukaryotes. So the Tata box is a combination of Tata, AAA. So it has Tata and AAA. So these are 1, 2, 3, 4, 5, 6, 7 nucleotides which are responsible to be one of the regulatory materials which is needed for the DNA for the transcription process in the eukaryotic cells. So this is the RNA polymerase. So what happens is the RNA polymerase is going to be toward the position where the unwinding process is supposed to start by the promoter. So the promoter will show the place, is going to point to the location on the DNA where the process of transcription should start from. So it's going to start the process of unwinding it from the location where the RNA polymerase forms a complex. So these are all the materials which is needed in the transcription process of eukaryotic cells. So the transcription is controlled by regulatory DNA sequences and the protein transcription factors. So most eukaryotes have what we call the Tata box promoter, and they also have an enhancer and the silencer. The enhancer will help to speed up, while the silencer will help to slow down the rate of the transcription. And each of these genes would have a unique combination of regulatory sequences. As you can see, there is a squeeze in here, a squishing of the DNA strand. The reason is because unlike the RNA, whereby all the materials which are needed for the process are being put together close to each other, but for the case of the eukaryotes, they're far away from each other. So this is the reason why it is being actually squeezed, it's actually being folded, so that all the materials which are needed for the process can be brought close to each other. And this is what you see here. So, and RNA processing is also an important part of the gene regulation in eukaryotes. You have to process it. So as you know, when the mRNA is being produced after the transcription process, which is going to happen here, there is something we need to know about processing the mRNA. How do we process it? This is the mRNA which has been made. Now it's a combination of what we call the exons and the introns. Exons are the ones that have the genetic material in them. They have the genetic material or the genetic codes in them. While the introns is known to have nothing, we believe the introns are just intruders, which are just coming to cause problem in the mRNA sequence. So that the reason now what we're going to do is we're going to slice it. We're going to use a slicing method to remove out to remove the introns, because the introns have no genetic materials in them and they have nothing in them, but they just increasing the length of the mRNA. Though some scientists have said that they just stay in between the exons so that genetic materials would not mix with each other, would not be transferred into each other. I believe that is not true, but hopefully we don't. To now, the scientists could not describe completely what the introns, what they do. Well, we just call them intruders, which are in between the exons. So once they've been removed, then we will process the mRNA by bringing them close to each other. And while we're doing that, we're going to put a cap in the beginning of the mRNA, and we're going to put a tail at the end of it. So to protect the genetic material which is in the mRNA. So this process is very important before the mRNA is being translated to form a protein. So it says the mRNA processing includes 3 major steps. Introns are removed and exons are being spliced. So the splicing method is the removal of the intron and the splice and put in the head and the tail for the exon. So a cap has been added and a tail is been added also. So this is the completion of the gene expression and regulation. Thanks everybody for listening.